Assalamu alaikum wa rahmatullahi wa barakatuh. My name is Faisal Alkhwaiter, and today we'll be discussing disposal of nitrogen. So, without further ado, let's get started. So, for our objectives today, these are the objectives we'll be talking about. So, we'll basically discuss what nitrogen balance is, and then we're just going to discuss what happens to an amino acid in our body after we digest it. So, just a quick question: can we store protein in our body? The simple answer would be no. Unlike fats and carbohydrates, amino acids cannot be stored. Why is that, you might ask? Let's discuss it in this slide. So, as you can see, when we consume protein, protein is very essential to our body. It's almost the building block for a lot of processes. So we do degrade it a lot and desynthesize because we cannot get the perfect amount of protein in every meal. So we need to improvise. So how do we do that? We basically ingest protein. We do already have our protein from our muscles and organs, which contribute to what we can see here as our amino acid pool. And depending on our needs, we can either break down protein or synthesize protein. And then the excess protein that we don't need, we can dispose of it as in urea and in urine. So just a very important point to point out is that in a normal adult or human, the nitrogen balance should be 0, meaning the protein that comes out is the same number of protein that comes in. So that is nitrogen balance. As you know, not in every circumstance would have a nitrogen balance of 0. We might have a positive nitrogen balance, meaning that the person is storing more protein than they're excreting. That's basically when you're trying to get nourished. So basically, in childhood, in pregnancy, we're actually building something. And we have, on the other hand, a negative nitrogen balance, which basically means we store less than what we excrete. So basically that happens when you're under trauma, when you're sick, you cannot synthesize proteins very well, so you just lose a lot of them. Or basically you don't ingest enough protein in malnutrition. So just a quick checkpoint. Excess protein cannot be stored in the body. The nitrogen balance of an healthy adult should be 0, and a person that just got out of surgery should have a negative protein, a positive protein balance, because I'm sorry, it's a negative protein balance because he just got out of stress. So basically, someone cutting into you is very stressing to the body. You'll lose protein. But if the question said someone that's recovering from surgery 3 weeks later, you'd probably be in a positive nitrogen balance. طبعا minding the diet that he's in. So let's talk about how do we transport amino acids in our body. So let's see myself. I just ate a couple of shawarmas for lunch, full of chicken, which is full of protein. After I ingest it, food goes to my stomach. We have a hormone called pepsin, which breaks down the dietary protein into polypeptides. And then further on, as you've taken GI already, you'd have duodenum. In the duodenum, there are important enzymes, digestive enzymes that are produced, which are pancreatic enzymes, trypsin, chymotrypsin, and so on and so forth. There are a lot, which break down the polypeptides into oligopeptides. And then further down, where it extends the small intestines, we do have proteins that are which are called aminopeptidases, which basically break down the oligopeptides into amino acids. A very important point to point out is that even when you have the digestion happening in the stomach, it doesn't break down into perfect order. So we don't have dietary protein, then everything becomes polypeptides, and then every polypeptide becomes multiple oligopeptides and so on and so forth. No, even through pepsin, we can have the breakdown of polypeptides and some amino acids that are already broken down. That's a very important thing to point out. So we just understood the normal. What happens if that process does not go well? So we have 2 pathologies that we're going to talk about. The first is going to be cystinuria. From its name, cysteine, the amino acid, and urea. So basically, we have a problem in uptaking the amino acids, which are called. So we have cysteine, arginine, ornithine, and lysine. Remember, thing we're going to take is probably in the next lecture after this, which is urea cycle. It's very important. And in regards of this pathology, we have problem in the proximal tubules of the kidney, which basically we cannot absorb cysteine, which is going to make us lose cysteine in urine. So when we lose cysteine in urine, that's not the biggest problem. The biggest problem here is that the cysteine that accumulates in the kidneys and the ureters may form crystalline stones, which can basically affect or cause kidney stones. A minute yet important point you need to know is that type 1, there are 3 types of cystinuria. Cystinuria 1, we'd have a normal amino acid profile, especially in the urine, in the urine. And then in type 2 and type 3, you'd have cystinuria, which is basically elevated levels of cysteine. The other pathology that we have is Hirschsprung disease, which is basically the defective ability to absorb tryptophan, basically in the stomach and the small intestines and the kidney. We cannot absorb this amino acid, and this amino acid is quite essential because it's the precursor of niacin, B2, which is a precursor of a lot and a facilitator of a lot of reactions. So the patients that have a deficiency in tryptophan will present with pellagra-like symptoms. So I'm pretty sure you took or touched upon pellagra, which it's usually characterized by 4 Ds: dementia, diarrhea, dermatitis, and then death. There's just a small point that I like to point out. Basically, when there's the defect in uptake of the amino acids in the kidney, there will be a defect in reabsorption. A defect in the small intestine will be a problem in absorption. And I think that's obvious from GI and renal, but it's just a good point to point out. So a quick checkpoint. Let's review. Pancreatic enzymes break down polypeptides into. It's actually both oligopeptides and amino acids. As we said, the breakage is not perfect. We may get amino acids directly from the interaction between the polypeptides and the trypsin, chymotrypsin, which are pancreatic enzymes. Increased levels of cysteine in urine is seen in cystinuria type 2 and 3. Type 1, the urinary analysis would be normal in regards of protein. In Hirschsprung disease, they'll have pellagra-like symptoms, such as we said, patients with Hirschsprung disease come with 4 Ds: dementia, diarrhea, dermatitis, and death. I know that diabetes sounds close, but no, it's not one of these. So let's hop into the next. So we do have a protein. We don't need it anymore, or it's damaged. We want to dispose of it. We cannot just take it and pick and throw it to the trash. No, we need to mark it and to our body to digest it. So how do we do that? We have something called ubiquitin, a very small protein which just tags a protein for it to be destructed. So what happens is we have the protein that we want to break down. We have our ubiquitin. If it sees that this protein is defective, it just attaches on it. And then something called a proteasome will just be roaming around and see, okay, you have ubiquitin, I'll eat you. You don't, I won't. So basically that's it for cellular for protein degradation at this point. So let's take it down to the molecular level. We have proteins. We have which we made into basic structures of amino acids. And now we want to utilize these amino acids for energy, for building. And how do we do that? Basically, this is the structure of our amino acid. We have our alpha amino group, we have our alpha carbon, and then we have our carboxyl group. Basically, this structure, the alpha amino group, protects the amino acid from getting damaged. So, and this part is usually called the carbon skeleton in regards of this. So this is basically the carbon skeleton, which can be transformed into other types of energy as intermediates for other things such as glycolysis, gluconeogenesis, and the TSA cycle. So for us to produce energy, we need to remove this bad guy here. It's not a bad guy, it's just we need just to remove him. So when we remove him, we produce something called ammonia, which is quite dangerous. Let's see how we would deal with such a situation. So as you can see here, we have protein, basically what I said, we have protein, amino acids, and then we'll end up NH4, which is urea. We'll discuss that inshallah later. And then we'll have keto acids, which can be metabolic intermediates in other reactions. So what happens when we want to try and remove, how do we remove the alpha amino group? We do it through two reactions, which are transamination and oxidative deamination. Let's take one step at a time. So what's transamination basically? Transamination is an exchange program. So we have our amino acid and we have our keto acid, which is alpha ketoglutarate here. It goes through the reaction, which is an aminotransferase. From its name, it transfers. So it's like an exchange program. It takes the amino acid, the amino group, sorry, and transfer it into a new structure, giving us a new amino acid and a new keto acid. So in the two reactions that we'll be studying, we'll have our amino acids, the two amino acids we're going to discuss now, and we have alpha ketoglutarate. They're gonna go in and produce a keto acid, which can be utilized in other reactions, and they'll give us glutamate, which is very, very important. So the two reactions that we're gonna discuss are ALT and AST, alanine aminotransferase and aspartate aminotransferase. As you can see here, we have alanine, and then we have alpha ketoglutarate. They'll go through the transamination reaction facilitated by PLP. PLP, or paradoxal phosphate, is the active form of B6 in our body. So basically, it utilizes this reaction, and then we'll produce pyruvate, which is ding glycolysis, and then we have glutamate. We can utilize the pyruvate in reactions if we need energy, or it can go down the line and we store it as fat. And then we have aspartate aminotransferase. We'll have aspartate here and alpha ketoglutarate. It will go through the transamination reaction, and it will get us glutamate and oxaloacetate, which is another intermediate of the TSA cycle. Basically, how do you differentiate? I like to think of alanine pyruvate because mostly alanine is very associated with muscle. We're going to talk about it in the alanine cycle. And I mean, that's how I know it, but I think there's a mnemonic for it. I'm not sure. It's two biomarkers are pretty easy. And ALT and AST, I'm pretty sure they ring a bell. They are biomarkers when you detect liver disease and sometimes heart disease. I think you took it in the enzymes lecture, but they're very, very important in diagnostic blood tests to check for liver function, because all of this happens where? In the liver. So for oxidative deamination, it's basically you take our glutamate and we use either NAD or NADPH. It is the only reaction in the body that could use either of those two intermediates and could yield us alpha ketoglutarate. A very, very, very important point that I forgot to mention is that these reactions goes both way. So if we need alanine for some reason, we take pyruvate glutamate and the reaction goes the other way. Same thing for aspartate and same thing here. So we'll use NAD or NADP, for us to if we need glutamate. And just a very important point to know, this might yield us NH3, which is ammonia. For a quick checkpoint, NH3 or NH4 is very toxic and needs to be excreted. Basically, it's ammonia, which is NH3. Transamination, we remove an amino group and donate it to alpha ketoglutarate to form glutamate. Glutamine is going to be very essential on the point we're going to talk about next. And then we have oxidative deamination, which glutamine dehydrogenase utilizes NAD and NADP as cofactors. This is very crucial because the only enzyme or reaction that utilizes that through that enzyme, which is glutamine dehydrogenase. So what does that mean in the bigger picture? Basically, as a person eats, so as I said, I've eaten the 2 shawarmas in the beginning, I have a lot of glutamate in my body. So what my body is, utilize the protein it needs and takes the excess protein, breaks it down, and takes the intermediate that we need through either usually fat storage in a fat state, and any excess metabolites that we don't need is usually secreted by urine. And for when you don't we do not have, so let's say I'm fasting in Ramadan, I slept through Maghrib and Fajr, so I woke up the next day and I'm still fasting. I cannot break my fast, so I'm hungry for 2 days. I haven't eaten. My body is at a low energy state. We would still have high degradation, but we will use the intermediates, which are in those two reactions, pyruvate and oxaloacetate, as energy forms to produce energy from these carbon skeletons that we derived from our amino acids. So now we might break down in muscle, these reactions happen. So if you have excess glutamate in the muscle and we want to transfer that so we can dispose of ammonia, sorry. If you want to dispose of ammonia, that every reaction that in regards to the urea cycle happens inside the liver. So how do you transport it through the body? We utilize something, the other amino acid called glutamine. So we take glutamate and then add ATP to it and add another ammonia, and then that will yield us glutamine. Glutamine is a non-toxic carrier of ammonia. It will move the glutamate, which we merge to make glutamine, to the liver, and then in the liver, the reaction would happen again, which is derived from the enzyme glutaminase, which will yield us glutamate back, and then that can either we can dispose of the nitrogen there, and then or we can also break down the glutamate again to produce alanine, or use the metabolites in the middle to take onto the muscle again to produce energy. Here, as you can see here, you have pyruvate, glucose, pyruvate, ALP, alanine, and then alanine, pyruvate. So it's a cycle. It's called the glucose alanine cycle. I think the doctor didn't touch upon it a lot, but it happens between the muscle and the liver, so we can utilize the energy more basically. And as I said, ammonia is detoxified in the urea cycle, which you'll take up on next. Inshallah, I believe those two lectures are pretty easy if you understand them from the bigger point down. So this is basically what happens. We have our reactions, which can yield us which you can dispose of nitrogen here in the urea cycle, or that could yield us the intermediates that we need for either energy production or fat storage. So we're done. Let's do a couple of questions to review some of the concepts. These questions I got from the textbooks that were recommended by the course, and they're very easy to answer, inshallah. Let's take them one by one. The waste nitrogen from skeletal amino acid catabolism is derived from the liver and the incorporation of the urea in which of the following? Okay, I do have the answers for them. I do believe. Wait, what happened? Yeah, it's alanine, so it's ALT. Sorry. Yeah, it is ALT. I thought I put the answer wrong. It is ALT. So basically, what happens is we have our transamination reaction, and after that we have our glutamate. We produce a glutamate which goes through glutamine and goes back to the liver as glutamate, and then the glutamate can excrete the excess ammonia, or can get back again into ALT and AST, which can yield us to the alanine. So which of the following represents the major compound of the circulation responsible for the transport of nitrogen? We want to transport nitrogen, or ammonia, sorry, we use glutamine. Glutamine is the main transport point. So basic true or false question. Vitamin B5 is critically important in the overall process of amino acid transfer from amino acid to a keto acid. That is false, because we utilize PLP, which is the active form of B6. Thank you, everyone. I hope this was fruitful for you. For the ones watching the recording, even you, please use the following QR code to rate the session for us to be better tutors in the future. That's it on my end. If anyone has any questions, don't hesitate to text me or email me under questions under this topic or in general in medical school. Thank you and have a great day.